This is presentation number 4, cerebral blood flow. Again, these talks are in a sequence. We build on some of the language and some of the concepts. We'll run through the questions, the way they're asked, run through some background information, and then we'll hopefully present a model answer. The ways that these questions are asked are quite variable. They are trying to make something technical out of what is really a very simple issue. For example, if we talk about cerebral autoregulation physiology, the STEM is that a 75-year-old man presents following a collapse. His GCS is 6 of 15. Start with physiology. What factors affect cerebral blood flow? What is meant by the term autoregulation of cerebral blood flow? Really have to draw a diagram. What is the Monroe-Kelly doctrine? That seems to be a popular way of asking a similar thing. Other ways that it can be asked. What factors affect cerebral blood flow? Which is again the same question really. What is the mechanism of the Cushing response? Again, regulation of blood flow, autonomic regulation. What is the Monroe-Kelly doctrine? Same thing again. It's got a fancy name, but it's really a simple concept. Other ways that it is asked don't vary a great deal. They tend to repeat these questions quite often. So the big things here are the Monroe-Kelly doctrine, is autoregulation, and factors governing blood flow in one way or another. So the explanation here of cerebral blood flow, namely autoregulation, is a pretty simple one. I'll assume that we know that flow times resistance equals pressure. We all know that mean arterial pressure is cardiac output times total peripheral resistance. It's the same way of writing the same thing. So if we talk about building this answer based on things we already know, pressure is flow times resistance. And the surrogates to that we know as MAP. We report MAP all the time, mean arterial pressure. Flow, cardiac output, the holy grail of trying to measure something non-invasive. When we talk about total peripheral resistance, this is when we talk about overall cardiac performance. We know these things. Believe it or not, all we're going to do is talk about an organ system, and all the same rules apply. Going to define a few things here. Flow, we all get the concept. If you take an artery, cut it, blood spurts, that's a flow. It's movement of a fluid. It's measurable and usually has units of mils per minute, mils per second. In fact, litres per minute, we know for cardiac output. If we talk about resistance, we're talking about a concept which is a little more difficult to grasp. I'll explain that in a little more detail in just a moment. Pressure, what we're talking about is the pressure head or the driving force on the fluid. And in reality, that pressure is a difference of two pressures. In our circulation, we talk about the difference between systolic pressure and diastolic pressure, plus 1/3 of that, as our map. What we're talking about is an arterial pressure minus a venous pressure. So for us to have a driving pressure on something, we need a gradient. And so I'm going to introduce the concept now of a pressure gradient. So really what we want here is the pressure gradient. And in our circulations, we talk about arterial pressure and venous pressure. And the difference between those two is what the organ sees as its driving pressure. So if we have a brain and it has blood going in, blood going out. The blood going in comes in a blood vessel. That blood vessel passes through the brain parenchyma. It's got to get into this black box called the skull. And in this black box sits a few other things. There's some more fluid called CSF. There can be pathological things that sit in there. A big ball of blood, a big tumour, other things that take up volume. And then we have the brain parenchyma. That's essential. Must be there. So, arterial pressure drives blood in, and venous pressure resists the outflow of blood. So, the pressure gradient is then the difference between these two. Pretty simple concept. Then what we can talk about is the flow, and that's really what's important. That's how nutrient gets to the tissue. Without enough flow, no matter what its pressure, there is no brain. It requires metabolism. And then we talk about resistance. Resistance is the multifactorial thing for the brain, and they like to ask this question as opposed to the kidney. They like to ask this question because there's a few other factors that come into it. The resistance you can think of as the compression of this blood vessel as it flows through the tissue in the brain parenchyma. That should be relatively fixed. You can also think about the resistance on this vessel as it goes through the vault or the skull, the rigid box, such that compression at that point provides a resistance. You can also think about resistance due to CSF, such that the resistance of the CSF pushes in against that blood vessel, providing a resistance. All these factors will resist flow at each stage. We can also now think pathologically and think of our tumour, our large ball of blood, as another factor that can dent this blood vessel and cause an increase in resistance. So all of these factors can change resistance, and they like to ask the cerebral blood flow question because we can talk about resistance of vasculature. We can talk about resistance due to tissue volume. We can talk about resistance due to fluid volume, CSF. Obviously, if you increase the CSF volume, squash the blood vessels, more resistance. Increase the tissue volume, here and here. We squash the blood vessels, and we increase the resistance. If we squish the blood vessels by occluding them here, or we can apply adrenaline to them, or we can, during a resuscitation, we can apply carbon dioxide to them if we ventilate inappropriately. All of these factors will change the resistance. So these are the areas of concern. Now, this all comes about because we have this black box called the skull, the vault, and it's not distensible. There's a few holes in and out of it, and if pressure gets too much, toothpaste starts to come out of the holes, i.e. that's bad for brain parenchyma. Loss of function, compression of various structures occurs on the way. So what we talk about now is this thing called the Monroe Doctrine, Monroe-Kelly Doctrine. Kelly, I.E. And this is a doctrine that just relates a fixed volume of a skull, and that's all it's about. So it's a fancy way to ask a very simple concept. Can you tell me how this relates to the Monroe-Kelly Doctrine? Well, yes, you can. You can say that the skull has a fixed volume, and it can only be taken up by a few things. Those things contribute to resistance and therefore blood flow. So they can only be taken up by tissue, by blood, and by CSF. That's our Monroe-Kelly Doctrine. So, when it comes to trying to work out what determines cerebral blood flow, these are the only factors. We have just written it out. Now what we want to do is to be able to present that in a way that we can draw a quick graph, get the concepts across, and show the examiners that we know what's going on. The relevant graph, again, can be put on a set of axes, and it relates flow and relates pressure. And you'll notice some similarities here to other graphs that we've been drawing. That's why these are in sequence. Again, we could be talking about cardiac curves, but here we're talking about just flow and resistance relationship for a particular organ. Equally apply this to the kidney. Equally apply this anywhere else. Everyone understands the concept of flow through a rigid pipe. Now I'm going to put that on here, first of all, as a reference point. Flow through the rigid pipe, by definition, has a fixed resistance. The resistance of the pipe doesn't change. Its calibre is what determines its resistance. We can force more and more pressure through the pipe, but we're not going to distend it. It's not going to contract. We can put a weight of blood or tissue or CSF on the outside of it, and it's still going to remain a rigid pipe. Its resistance doesn't change, i.e., its slope doesn't change throughout its length. The slope of our rigid pipe, a rise over run, is always the same. It's fixed. Very simple concept. Unless you're 100 years old, full of vascular disease, we don't have rigid pipes as blood vessels. What we actually have is distensible organs, and we also have a black box to deal with. If we have too much blood inside the skull vault at any one point in time, we're going to end up with an increased pressure. It stands through the Monroe-Kelly doctrine that the overall volume is fixed, and if we increase the proportion of one thing, we've got to decrease the proportion of the other. To create too much pressure in here increases our pressure gradients, increases our resistance, and what we're trying to do here is defend flow, defend brain metabolism. So to allow too much blood flow into the brain immediately impedes something else. Everything else in there has a function, exclude our tumour. So it's important for the brain to not have too much flow, i.e. too high on this curve. But it's also important that if we have a decreased blood pressure, it's important that the brain still receives a lot of flow. It takes a large proportion of your cardiac output, and it must, 3 minutes, and it starts to damage. So what we want to do for the brain is maintain a constant flow. Now, if I was to use this concept to demonstrate what that looks like, we would say that over a range of pressures, we would have a single constant flow, i.e. a range of pressure, one value of flow. In fact, that's what we're trying to replicate. It just so happens that evolution would say those who couldn't regulate that in that way died out. So we now end up with a situation where, for the brain, we have flow that remains the same over this broad pressure range. Now we're going to put some limits on that. The way that we make that adjustment to flow is to make our blood vessels not behave like rigid pipes. I.e., the rigid pipe, there has to be a portion of rigid pipe. Physiologically, it must exist. But then we're able to exert some change or some influence over our vascular structures in the brain. So what we do is we change the resistance. The rigid pipe can't change its resistance. Cerebral blood vessels are going to change their resistance. So what we'll do is allow them to dilate. In fact, the resistance will tend to go down compared to the rigid pipe. So if we allow them to dilate, we end up with a curve that maintains flow fairly flat by allowing the blood vessels to dilate up a bit, or allowing them to contract, or both. So we're not a rigid pipe, but there's obviously a point where if we allow blood vessels to dilate up, we get too much blood in the brain. So there's got to be a happy medium somewhere. What we're now going to do is show that other end of the curve that instead of maintaining constant flow, there has to be a limit. We can't dilate blood vessels infinitely. They get to a point where they behave like lead pipes again. In fact, they behave a lot worse than lead pipes, and they tend to do this until they break. So what we have physiologically, you can verify with experiments, is a brain that has the ability to maintain a constant flow over a range of pressures. It's obviously going to sit at around about 100 mean pressure, mean perfusion pressure, so that it's got a large range of pressures either side that the brain might be subject to regulate that flow. If we have too low a pressure, we're going to fall off this curve, i.e. act like a lead pipe. If we go too high, we're going to jump off the curve again and end up behaving like a lead pipe, in fact worse. So, for this to occur, resistance has to change. Flow over pressure. Sorry, pressure over flow. So, when we change our resistance, we're behaving less like a rigid pipe, and we are able to fend off the imposed changes of flow that are changing pressure or increasing pressure we produce. So, where we now get to is the changes that occur with changing pressure in the cerebral vasculature. So if you can imagine you increase your pressure and flow is to remain constant. If you increase pressure to keep flow constant, we've got increased resistance. If this value goes up and that one remains constant, that has to go up. We increase resistance on our blood vessels by constricting them. So this is the constriction end of the curve. If pressure falls, conversely, we dilate our blood vessels. This is the dilation end of the curve. This phenomenon of the blood vessel looking after its own flow is called autoregulation. So that particular organ autoregulates. And that's where that word comes from. That's what it's describing. The ability of the tissue to regulate its own blood flow. And there are various mechanisms, depending on the organ, that control this. Most notably, in the brain is the myogenic response. It's an innate, metabolic-driven vasoconstriction and dilation of blood vessels in response to flow. In response to stretch and pressure, which governs flow. So what we have here is autoregulation of the cerebral circulation. Over a range of pressures, flow is held constant. Just so happens, we can define these pressures. And we talk about, roughly, for a given brain, between 40 and 150. Mean pressures, mean perfusion pressures, differences between arterial and venous. And we tend to regulate blood flow over that period. So what we're talking about is trying to adjust the lead pipe and its resistance to facilitate blood flow. These questions are also answered not just in relation to cerebral blood flow, but also other tissues. It's very important that the kidney maintains its own blood flow. It has afferent and efferent controls in the circulation to maintain a glomerular pressure. You can also see that if we use another tissue, such as a skeletal muscle, it's a large volume and can be used to regulate blood pressure. And in fact, we see this type of response in skeletal muscle, such that pressure is maintained relatively constant over a large range of flows. Skeletal muscle changes flow to regulate pressure. It's one of the main determinants of your baroreflex. That's how all of this is related to stuff that you already know and you've already read. What we're going to do now is relate it all back to Monro-Kelly doctrine. So the physiology viva that deals with cerebral autoregulation will be asked in the STEM as a 75-year-old man presents following a collapse. His GCS is 6 out of 15. We'll start with physiology. First question, what factors affect cerebral blood flow? Cerebral blood flow, like all organs, is related to flow times resistance. If we're talking about cerebral blood flow, we're talking about factors that influence the pressure gradient, such as the pressure in the arteries upstream and the pressure in the veins downstream. So venous pressure and arterial pressure are direct influences of that. If we're talking about the cerebral resistance, we're talking about vascular tone, we're talking about CSF pressure, we're talking about brain tissue volume or brain swelling. We're also talking about external factors that can produce changes, such as pathologies of tumour or mass. The second part of the question is what is meant by the term autoregulation of cerebral blood flow. Autoregulation defines the ability of an organ to maintain its flow at a constant rate in the face of changing pressure gradients, diffusing flow. For the cerebral circulation, we're talking about rough limits of between 50 and 150 millimetres mercury, corresponding to a constant flow over this period. This is accomplished by the mechanism of autoregulation, which relies on the myogenic mechanism innate to blood vessels. They feel stretch in the face of increasing distension caused by increasing pressure. The third part of the question is what is the Monro-Kelly doctrine? The Monro-Kelly doctrine states that for a brain sitting inside of a skull, that the volume of the brain tissue, the CSF, and the blood that bathes it is constant, such that a change in the volume of the blood in the skull causes an increase in pressure. Likewise, an independent change in the CSF volume changes pressure. An increase in CSF volume, such as hydrocephalus, increases intracranial pressure. Likewise, an increase in cerebral tissue volume, such as diffuse cerebral edema, causes an increase in pressure. All of these factors, if they increase pressure, compromise cerebral blood flow.